Good day, everyone. Today we will present you our video presentation of Science, Technology, and Society. Our report entitled Genetically Modified Organism. But before that, let me introduce you our objectives. Our objectives, at the end of the topic, the readers are expected to define genetically modified organism, explain the principle behind cloning and genetic engineering, answer frequently asked questions about GMO, give examples of GMO plants and animals, discuss the advantage and disadvantage of GMO. What are GMOs? GMOs are plants, animals, or microorganisms whose genetic material or DNA has been altered using genetic engineering techniques, such as inserting a specific gene from one organism to another organisms. Introduction of our report. The genetic modification of organisms is a vast industry, and the application of the technology and existing and far-reach, it brings new hope for medical cures, promises to increase yields in agriculture, and has the potential to solve the world population and resource crisis. Organisms with artificially altered DNA are referred to as genetically modified organisms or GMOs. Molecular biologists have discovered methods to change the structure of DNA in living organisms and alter the way in which the organisms develops. However, the risks of using GMOs have been the subject of considerable debate in recent times. Most experts accept that provided GMOs are tested carefully, the health risk to humans should be minimal from plant products. Although the health risks from animal GMOs are potentially more serious, especially when the animals are for human consumption. The potentially huge benefits to the gain from the use of GMOs creates enormous pressure to apply the existing technologies. However, there are many concerns including the environmental and socio-economic effects and problem of unregulated use. Key concept of genetically modified organisms. Genetic engineering. The development and application of scientific methods, procedure, and technologies that permit direct manipulation of genetic material in order to alter the hereditary traits of a cell, organisms, or population, also called recombinant DNA technology. Genetic engineering, also known as genetic modification, it is the science of direct altering an organism's DNA to change its traits or functions. Genes. Basic unit of heredity in a living organisms. Genes come from our parents. We may inherit our physical traits and the likelihood of getting certain diseases and conditions from a parent. Genes contain the data needed to build and maintain cells and pass genetic information to offspring. Genes are the fundamental units of heredity, tiny segments of DNA that carry instructions for how living organisms grow, develop, and functions. Organisms. Organisms, a form of life composed of manually independent parts that maintain various vital processes, a plant, animal, bacteria, and fungi. Explanation of organisms. Organisms are any living entities that carry out life processes such as a growth, reproduction, and metabolism. They range from the simplest bacteria to complex beings like humans. Vector. Vector, a DNA molecule that carries the foreign genetic material into a cell so that the foreign genetic material can be copied or used by the cell. A vector is a DNA molecule, often a plasmid or virus, that acts as a delivery system to introduce foreign genetic material into a host cell. Once inside the cell, can replicate or express the new DNA, meaning it can make copies or use it to produce proteins. Plasmid, small circular pieces of DNA found in almost all bacteria and in some fungi, protozoa, plants, and animals. Extra chromosomal elements capable of independent replication and commonly used in plant transformations. A plasmid is a small circular piece of DNA found in many microorganisms, especially bacteria, and occasionally in other organisms like fungi and plants. It's separate from the chromosomal DNA and has the unique ability to replicate independently. Deoxyribonucleic acid or DNA, a large organic molecule that carries genetic information in the chromosomes, making it possible to transfer the genetic characteristic in all life forms. Resembles a twisted ladder. DNA, it is the molecule that carries the genetic instruction for life. It's like nature blueprint telling cells how to build and operate an organisms. Cloning is a technique or process used to produce genetically identical copies of a biological entity. For example, Dolly. Dolly is the first mammal to be cloned from the cell of an adult animal at the Roslin Institute in Scotland. Cloning is the process of creating an exact genetic copy of a biological entity, whether it's a gene, a cell, or an entire organism. It's like making a duplicate with the same DNA blueprint. So that is called cloning. Steps in cloning. Step 1. Cloning requires a source of DNA or donor organism. Step 2. The DNA is cut with restriction enzymes and is ligated to a vector. Step 3. The vector, usually a plasmid previously cut with another restriction enzymes compatible with the first enzymes used. Step 4, or the last step. After ligation, the chimeric molecules are transformed in competent cells and then screened for the presence of genes of interest. The process of cloning, especially DNA or gene cloning, involves creating identical copies of a specific DNA sequence. Gene therapy, genetic modification of human or so-called gene therapy, is becoming a treatment option for diseases ranging from rare metabolic disorder to cancer. Coupling system cell technology with recombinant DNA methods may someday allow stem cells derived from a patient to be modified in the laboratory to introduce a desired gene. Gene therapy explanation. Gene therapy is a medical technique that uses genes to treat, prevent, or cure diseases by correcting faulty genes. It works by introducing, inactivating, or replacing a disease-causing gene with a healthy version to restore normal function. Gene therapy can be delivered either by removing cells, modifying them in a lab, and returning them to the body, or by delivering the genetic material directly into the body.